I am pleased to have Dr. Brian Ogilvie here with us today. Brian received his Ph.D. in toxicology from the University of Kansas Medical Center. He has been with XenoTech since 1997 and currently serves as the Vice President of Scientific Consulting. Dr. Ogilvie is an author or co-author on over 50 scientific posters, peer-reviewed publications, or book chapters on the topics of drug metabolism, transport, and drug-drug interactions, and represents the company as an invited speaker at various drug metabolism conferences through drug-drug interaction-related R&D projects and expert opinions for consulting clients. It's my pleasure to present to you Dr. Brian Ogilvie.
Thank you, Michael. And so the title of my presentation is Essential Considerations on the New FDA in Vitro DDI Guidance, the what, the why, and the wow. So for today's topics, I'm going to start with an overview of the major changes to the guidance from 2012, why some of these changes were made, are these recommendations or requirements, and then the worldwide DDI guidance landscape, and why science is always a good trump card, and an overall general strategy to progress candidate development in the face of these new guidance documents. I won't be able to cover some topics, for instance, the PBPK. So the two guidance documents, I've highlighted the in vitro one because that's the one I'm going to be focusing on today. I will mention the clinical drug-drug interaction guidance just at the beginning because it covers essentially the timing for these studies. And you'll notice that both documents say this guidance document is being distributed for comment purposes only. I wouldn't necessarily take that literally. I would assume that this is already the agency's current thinking, and I'll go through some of that in detail. So together, these are about 80 pages. 25,000 words. So there's no way that I'm going to be able to get to all of it in less than 1 hour. I'll assume most of the audience is familiar with the 2012 draft guidance, and I'll focus on the main points and concepts rather than all the details, and there are a lot of details. There will be follow-up webinars. And my apologies in advance for when I read some of these phrases. It can be somewhat difficult to actually paraphrase guidance and regulation, so I want to leave you with some useful information as in the form of a PDF later on as well. So the wow, I'm going to start with that. A few things that we didn't know were coming, at least from our perspective. So for instance, a requirement for validated analytical methods for reaction phenotyping. So these will be validated for purpose. For instance, is it just the parent? Is it the parent plus 2 metabolites? What matrix is it in, et cetera? There are some basic model changes. For instance, the R1, this is for direct inhibition. In the past, it was greater than 1.1. It's now 1.02. It looks worse. However, the good news is you can actually use the fraction unbound in plasma, but no lower than 0.01. So if you have anything that's plasma protein binding higher than 99%, you have to cap it at that value. The R2 cutoff, this is for the KNAC where you have true metabolism dependent or mechanism based inhibition. That cutoff is still 1.25, but it now includes a 50x unbound Cmax safety factor. The R3 cutoff, this is the one that we knew from before for the induction studies. The cutoff used to be 0.9, it's now 0.8. But it now has a 10x safety factor, which essentially penalizes low plasma protein binding drugs. So any drug with a fraction unbound in plasma of less than 0.1. So take, for instance, a drug, total Cmax is 1 micromolar, and the fraction unbound is 0.5. So 10 times that unbound plasma Cmax is now 5 micromolar, or 5 times the total Cmax. So that's something that we have to consider going forward, and especially for low protein binding drugs. So timing, this is something that's really changed in this guidance, and it comes from the clinical aspect, and I'll touch on that here in a minute. So basically, in a nutshell, you need more definitive in vitro data much earlier. This includes metabolism-based drug-drug interaction studies. Quote here, sponsors should initiate in vitro metabolic studies before first-in-human studies. And then even also to include some transporter data earlier. So if you look back at the International Transporter Consortium paper from 2013, I'm adapting a slide from that, going from discovery to first-in-human to proof of concept, and then proof of concept on to new drug application and marketing. In the past, you needed to consider the clinical program and which parts you wanted to de-risk. Most of these studies, the in vitro DDI studies, came, in this first-in-human to proof of concept, or even later into phase 3. With this new guidance, it's really suggesting you need to have these definitive in vitro data much earlier, considering indication, co-medications, et cetera. So really keep the clinical program that you have in mind early. So work backwards from the clinical guidance for the timing. When are the clinical DDI results needed? If you look at the clinical guidance, it now says before administration to patients taking the interacting drugs. So the quote directly from that is, inadequate studies of drug-drug interactions can hinder the FDA's ability to determine the benefits and risks of a drug and result in restrictive labeling, and this is particularly important, post-marketing requirements and post-marketing commitments, and or delayed approval. And I think everyone is probably concerned about any of those. Another quote directly from this is, to collect enough drug-drug interaction information to prevent patients from being unnecessarily excluded. If your exclusion criteria are such that it makes it difficult to enroll, it can really delay your program. So again, going on to the major changes now, the what. Earlier in vitro studies, as I mentioned, so this is one driver for the suggestion that test drugs be generally as high as possible for most studies. So because you won't actually have clinical data unless you've done some first-in-human studies, maybe in Australia or some other place, and don't have an open IND, you may not have clinical data. So this is a driver of using the highest concentrations possible. So that means solubility limiting, cytotox limiting. One silver lining to this is that it takes away the problem of only having data, in vitro data, up to the Cmax when your indication or formulations change as your program develops, and the Cmax is higher than what you had in your single ascending or multiple ascending doses. The Met ID, so this is strongly implied. It doesn't say it directly, but it really implies that these studies must be done first, because the metabolic pathway identification to determine whether and how to conduct reaction phenotyping studies. Most studies, you now have to have more than 3 replicates for everything, so you get the nice little standard deviation error bars. You need to show the selectivity and potency of chemical inhibitors. And I should mention that at XenoTech, we did publish a poster, ISSX in 2014, where we show the potency and selectivity of chemical inhibitors in reaction phenotyping studies at multiple conditions, all the way up to 1 milligram per mil for 2 hours of incubation, both in microsomes and in hepatocytes. So again, other major changes, validated CYP assays. We've been using validated CYP assays at XenoTech for years. CYP inhibition, the unbound inhibitor concentration must be shown or predicted in vitro. You can predict this if the log P value is less than or equal to 5. And if you've read the guidance closely, you'll note that it does not say that directly. But it does cite the Gertz et al. 2008 paper. So you need to be familiar with the literature cited within the guidance. We may need to evaluate individual isomers as perpetrators, and I'll talk a little bit about an example that we've seen just over this last summer regarding that. And then the transporters, mates have been added to harmonize essentially with the EMA, and I've got that here for the kidney proximal tubules. There are many other transporter details, for instance, multiple concentrations for substrate evaluations. I'm not going to go into those in today's webinar. For induction, major changes, once again, we can use activity, assuming that you know the inhibition profile. And then, as we've been recommending for years, if you have CYP3 induction, you should go back and look at evaluating CYP2Cs. And this is where activity as an endpoint is important, especially for CYP2C19. We know that you see very little mRNA induction, but you do see induction of CYP2C19 activity. And that can also be true for some of the others, CYP2C8 and CYP2C9. And this is one place where the guidance actually specifies, in addition to highest concentration possible, at least one drug concentration that's 10 times the unbound Cmax. And again, you don't necessarily have the clinical data, so this may have to come either later in development if possible, or just simply go with that general guidance to go as high as possible, cytotox or solubility limiting. You also have to evaluate the concentration in media. So this is similar to the EMA guidance where we look at what we call spent media analysis, and then also the unbound drug concentration. For induction, now when it comes to modeling, there are 4 basic models. In the past, we only had the one. This is similar to the EMA guidance where we have the fold change method. And it says greater than or equal to 2-fold change and greater than or equal to 20% positive control response. So there's some debate on whether this guidance really meant to say and, or if it should be or. If you consider something like CYP1A2, where you might get a massive induction with your positive control, but you could very easily have a concentration dependent 2-fold or greater induction by your test compound, then it would suggest that you don't have to do a clinical DDI study based on that. So if it's or, that would be a little bit more conservative. There are 2 correlation methods. There is the one that we know from before, the relative induction score, and then a new correlation method that some people have used in the past, Cmax unbound over EC50. And then what we know is the R3, the basic kinetic model. As I mentioned before, the cutoff value is now less than or equal to 0.8. Notably, there is no net effect model anymore, so you can't have a case essentially like the EMA. They have the same idea that you shouldn't offset mechanism-based inhibition with induction or vice versa. So for metabolites, there's a number of changes here, but in general, the evaluation of the drug-drug interaction potential of metabolites comes down to a risk-based assessment. You have to consider safety margins, concomitant meds, indications, etc. And then this is new. Phase 2 metabolites, it says that they're often better substrates or inhibitors of transporters than the parent drug, which is often true, but the transporter interaction potential of metabolites really is case by case, depending on, again, indications, meds, etc. When it comes to metabolites as substrates, this is also something new. So you need to determine if your substrate is a if your metabolite is a substrate of metabolizing enzymes. So if it's active, quote, if it has a potential to affect the safety or efficacy and contributes greater than or equal to 50% of the activity. And that is based on in vitro receptor potency and on in vivo exposure. But again, remember, because these studies are earlier and earlier, you may not actually have in vivo exposure. And then when it comes to metabolites as perpetrators, this got particularly difficult to put into a slide, so I came up with a flow chart. So first off, the guidance does say that if you have indications that you need to do a clinical drug-drug interaction study with the parent drug, generally you can stop there. You don't have to worry about your metabolites. It's assumed that the metabolites are evaluated in your clinical drug-drug interaction study. However, if that's not the case, or you don't reach clinically relevant exposures of the metabolite, then you need to do metabolite studies. And now there are some additional caveats. So for instance, if the metabolite is less polar than the parent and its AUC is greater than or equal to 25% of the AUC of the parent, or if the metabolite is more polar than the parent and its AUC is greater than 100% of the AUC of the parent, then you would need to do a metabolite study for that metabolite. An additional caveat, so if the metabolite has a structural alert for time-dependent inhibition and its AUC is greater than 25% of the parent, as well as 10% of total radioactivity, then you would need to do a metabolite study for that particular metabolite. The additional caveat here is if you don't have radioactivity data, then you only look at the greater than or equal to 25% of parent AUC as your criteria. So a number of things here. Since this is all before clinical studies, again, you may have to delay some of these metabolite studies until after clinical data are available and you know this. So this is something that we'll have to consider and may be something to put in the comment periods. So the why, we knew some of this was coming. In fact, this was listed in the guidance agenda on the FDA website as early as January 2016. I had heard from some of the FDA regulators that CDER had finished their draft of the guidance at the end of 2015, but there was a lot of internal review. So that appeared again on the guidance agenda for 2017, and as we know, now it has actually been released in October. And I want to mention that going back to 2012, lots of people spent a lot of time submitting comments. 43 public comments were provided, and don't think that just because the 2012 guidance wasn't finalized that these weren't considered. For example, one comment that appeared over and over was that the guidance document was difficult to navigate. So now we have that has been taken into consideration, and we have two documents to separate the in vitro and the clinical drug-drug interactions. We also have gone back to the appendices with experimental details suggested, similar to the 2006 guidance. And then there were lots of comments on harmonization between EMA, FDA, and PMDA. There were meetings between these agencies in March and May of 2017, and there were static models. Transporters were more harmonized, but I have heard from one or more EMA or FDA staff say that they simply couldn't agree on everything and that there will be differences. There were changes in basic model and cutoff criteria. For example, there was an evaluation published by FDA and other authors in 2016 looking at 6 methods for OATP 1B1 that minimizes the false negative rate and optimizes the false positive versus the true negative rate. Settle on one of those methods. And then similar analyses were published for PGP, OATs, OCT2, and the mates. And then also, some of you may know there were several publications and presentations between 2012 and 2017 from the FDA. And even as late as September 2017, there were 2 publications that really have many of the basic tenets that are in these guidance. So for instance, this publication published 2017 in September, and JPS covers the in vitro to in vivo extrapolation as well as the basic prediction models. Most of these are listed in the new guidance document. Same with the clinical. So this was published in the same issue, and it covers both the clinical drug-drug interaction designs and in silico, the PBPK considerations. And then there were several publications, not necessarily by FDA, but that were considered on long-lasting inhibition of OATPs that has led to the requirement for the 30-minute pre-incubation in OATP assays for inhibition. So cyclosporin, there were at least 3 from the period 2010 through 2015, and then more recently with PAWA and others, the dasatinib with 2017 just recently. And then we have seen some requests from the FDA to sponsors in the last year. So for instance, we had one client that was ready to enroll patients for an anti-cancer drug, and then they were told by FDA that they needed to have transporter and reaction phenotyping data prior to their first in human studies, or to add additional exclusion criteria. And these exclusion criteria would have been pretty restrictive and were relevant for that patient population. So they had to relent and go ahead and run these in vitro studies before they decided to start and enroll their phase 1 patients. And then we had a request from a sponsor over the summer that they had to evaluate individual epimers as perpetrators. So that essentially doubled the scope of their study, even though they could show that in incubation, if they started with either epimer alone, they reached equilibrium pretty rapidly, and if they started with the racemate, it stayed in equilibrium rapidly. So that was a request. Sometimes you can open a dialogue and push back on those things, but in the end, it may be easier to just go ahead and run the studies, and that was the case here. We've also had some requests directly from sponsors that we didn't realize would actually be into the new guidance. So for instance, individual positive control substrates for the recombinant CYPs and so on. In the past, we've used just a general P450 substrate to show that the recombinant CYP works and then run the assays. So on the question of are these recommendations or requirements, in general, I would recommend to expect to adapt quickly, but keep your programs moving. So, why do I say that? I think it's worth actually looking at the guidance regulations. These are not guidance documents. These are the good guidance practice regulations, which were revised in April 2017. So for instance, if you're not familiar with this is 21 CFR 10.115. And I'm going to quote a few statements from that. Are you or FDA required to follow a guidance document? The answer is simply no. But they do represent the agency's current thinking, and importantly, FDA employees may depart from guidance documents only with appropriate justification and supervisory concurrence. So any FDA staff would need to go to their supervisor and get approval to depart from that current thinking. And then when it comes to finalization, the FDA noted, or the regulation notes that the FDA also can, and I take that word can meaning might, review any comments received and prepare the final version of the guidance documents when that incorporates changes when appropriate. So in other words, don't wait for the final guidance. For example, 2006 and 2012, those were draft guidances and they never became final. And next I want to look at the worldwide view. So if you look at EMA, the adopted guideline was published in 2013. The concept paper was published most recently in 2017 in April. So the new the adopted guideline from 2013 is now considered to be under revision. And the work plan for the pharmacokinetics working party for 2017 included face-to-face meetings scheduled for October 2017, including ongoing liaison with the FDA. And so there may be some time here, but it's unpredictable as to when it would come out. So for instance, the proposed timetable in the concept paper notes that after receipt of comments, the draft guideline will be consolidated and released after 6 months of external consultation. But if you look at the previous timelines, there were a couple of years that elapsed here. So I think it's unpredictable. However, given that FDA has already released theirs, I'm hoping that this will be a shorter time frame. And one thing I should note is that, and I've heard this directly from some EMA reviewers, is that previous draft guidelines have not been considered to be in force. So then possible changes coming to the EMA guideline from that concept paper include induction study design recommendations, cutoffs for inhibition and induction of enzymes in the intestine by poorly soluble drugs, and a twofold cutoff for time-dependent inhibition. So this would be the IC50 shift, for instance. And then showing that you're maintaining the unbound target drug concentration during incubations. And then BSEP, we know now that the FDA guidance does not include BSEP. EMA does, and presumably will continue to do so, but now they'll provide more guidance on how to use that data. And then as far as Japan, the PMDA draft guideline was issued in September. This was closed for comments in October, and they're not made public. And there's also no publicly available English translation yet. There are some major changes, and we've been working with our colleagues to look at some of these. So for instance, decision trees for the necessity of clinical studies will be added to the appendix. There'll be lists of substrates, inhibitors, and inducers in the appendix, and also likely online, similar to the FDA. And then efforts are made to harmonize with the FDA and the EMA. So why good science is always a trump card, and I'm going to go through why I think that. These guidances describe, and this is a quote again directly from the guidance, a systematic risk-based approach to assessing and mitigating DDIs. So there are plenty of example statements on alternatives. For instance, the sponsor may justify alternative cutoff ratios based on its prior experience. And then on metabolites, for instance, alternative methods are acceptable if the sponsor can justify that the DDI potential of the metabolites can be adequately assessed. And they cite specifically these 3 publications. You can look within the guidance for those specific citations and find the full papers. And again, it points out that you have to be familiar not only with the guidance, but the citations that are within the guidance document. They also mention what they call proper or comprehensive justification for alternatives should be provided. And I think that publishing your own peer-reviewed data can provide very good justification. And case in point are these 3 publications on alternatives for looking at metabolites. So overall strategy, now that the new guidance is out, if you're in late clinical, basically my message is don't panic. Take all opportunities that you can to keep your communication lines open with the FDA. Go over your strategy, and you may be able to negotiate post-marketing commitments. Other candidates that are earlier that have definitive in vitro drug-drug interaction data already, I recommend performing a risk-based gap analysis for your in vitro package based on what you have versus the new guidance. And then depending on the development stage and your timeline, you may have to only reinterpret data based on new cutoff criteria. It may also be that you actually have to perform additional in vitro studies if needed for new drug applications. And we are actually actively assisting sponsors in this process right now. Basically, my message is to keep your drug candidate programs moving. We know that drug development is a lengthy process. Any delays can be quite costly. So we've already developed or have plans to cover most new designs that are in the new FDA guidance documents. So I'm going to leave it at that so we have time for questions, but I do want to acknowledge a number of people. This was a team effort going through these with a fine-tooth comb. And it's been, a lot of questions beginning with, which line numbers are you looking at? You have to read this very closely. So with that, I'd like to thank you for taking time out of your day or your evening, wherever you are in the world, and take any questions. So one question, for instance, do you have any new assay systems for the new drug-drug interaction guidance? So as I mentioned, we do have strategies for most of these, and we're working on the remaining few. Basically, we're using the comment period to develop an overall response, but we're moving as quickly as we can. Okay, so yeah, for the 4 basic models, let me go back to slide 12 real quickly. If you look at, again, because these are basic models, these are all just your first cutoff. So for instance, just as before we had the R3 value, if you can make that threshold, you can have strong arguments that you don't need to do clinical drug-drug interaction. And same with any of these other approaches. These are all just basic model approaches that you would use prior to going to the full-blown mechanistic static model, for instance. And let me go back to my final slide just so that anyone looking at the slides knows we're on questions. And then there is a question on slide 17. Was the transporter data required prior to first in human? It appears that this is a suggestion. I think, again, because there are alternatives that can be provided, you can provide those, but it does look like now the transporter information is being required earlier and earlier, And I say that because of my firsthand experience with the client that had a question from FDA, really a suggestion to include very restrictive exclusion criteria without those data. And another question about, has FDA mentioned anything about aldehyde oxidase specifically? And I was surprised to see that in the other enzymes section that was not listed. That was kind of one of my things that I intend to ask as a comment if we get the opportunity. And then the timing of substrate versus inhibition of transporter substrates, the same before first in human. I think you can develop your own strategy on that. So the guidance does say that you do need to look at a substrate evaluation for both PGP and BCRP for most drugs. They give you one out, which isn't much of an out. That's if you've proven your drug is a BCS class 1 using the biowaiver guidance. So that's a pretty high bar. So in many of these cases, it may be easier to just go ahead and run some of these in vitro substrate assays earlier. And I would mention that Ayman El-Khattan, he's been publishing on the extended clearance concept for quite some time, and has noted that the old idea of looking at transporter interactions in phase 2Bs or later is probably antiquated. We need to look at these earlier and earlier, and now the guidance seems to be suggesting that. I have a question here about if you measure the concentration of drug in a hepatocyte media and it changes over time, what concentration do you use to estimate EC50 for induction? So this is, I think, something that you have to decide once you have the data. So you look at the concentrations that you see on the last day of dosing, and if it does change over time, there are multiple ways to look at the average concentration on that day of dosing and use that. You can come up with an approach that corrects the EC50. We have not developed that further enough to really describe it in detail, but that is something that we are looking at actively. Another question on induction of CYP2C19, saying whether or not it has to be activity and mRNA. In that case, I think you can go, because mRNA is easy to just simply harvest when you run the assay. You could also go back and look at CYP2C19 mRNA later on. However, it's not going to be as informative, I think, as collecting CYP2C19 activity. So that's something that you would need to decide in your strategy for the study. Sorry, we have one more question. Comment on the new formula for the hepatic concentration, which now includes the RB value, the blood plasma ratio. So if I remember correctly, that blood to plasma ratio was included in the past, and generally what most people did was they assumed that it was, essentially what it would be based on hematocrit. So we are seeing more and more questions about actually, and requests to determine the blood to plasma partition ratio. So I think that does need to be accounted for in many cases. Another question about the time-dependent inhibition assay, should we use dilution method or no dilution method? So we've commented, well, we published on this extensively in the past, and for the initial experiment, our recommendation is still, when you're looking at IC50 shifts, not to use a dilution, and you just compare the shift, and then if you do have a shift, go on and look at K and ACT determination, where you do in fact include a large dilution factor. So then there are general questions about some of the studies that have been done in the past and whether or not they need to be repeated. I think again that question goes to the overall strategy. Do you need to consider what stage of development you're in? If it was years past and you're already into phase 3s and it's something on a minor criterion, something like now you have to include 2 transporter inhibitors, for instance, or maybe even the 30-minute pre-incubation for OATPs. We have always included a 15-minute pre-incubation, so I don't know if there's really strong data to suggest that 30-minute is much better than 15. I think it was just a compromise on settling on a 30-minute pre-incubation. So again, these are all case by case, and we are actively dealing with questions in continued conference calls, et cetera, with our clients right now to develop strategies for some of these issues. Another question, are the cutoff values closer between the FDA and EMA now? I think it's more of a compromise. That said, we don't know what's in the EMA guideline, and so that potentially could change. But I think the idea was, from hearing from Shumei Wang and others, Ping Zhao, et cetera, there were attempts to harmonize and look at an analysis of different methods that, as I mentioned, were published in 2016. So another question, FDA does not specify whether Ki for time-dependent inhibition should be used based on total or unbound concentrations, and what does XT use? So that, if I remember correctly, has the 50-fold safety factor for the K big I based on the Cmax. Now, whether or not you're talking about the K big I in vitro, I think going forward, we're going to have to more often correct for the in vitro value so that you have both sides of that. You have the in vitro correction as well as the in vivo correction for plasma binding. One more question on, is time-dependent inhibition using hepatocytes better than microsomes? I don't know if it's better. I think it is what it is. You know the limitations of your systems. You can say that, if you have a drug that is not avidly taken up into hepatocytes, or is avidly taken up into hepatocytes, then you might have a very different concentration in the hepatocyte than you would have in your microsomal incubation. So depending on what you know about the compound, you may strategize to use a different test system. The best way to determine Ki and Knac for time-dependent inhibition, the numerical method or the replot method. So there are quite a few approaches out there, and I don't know if I want to comment on what the best one is. I haven't done that sensitivity analysis. I think it depends. You would have to actually perform a sensitivity analysis to determine that. I think there are some other statistical methods to determine what would be the best, and then it's a matter of reaching consensus. I think the methods that we have in place are perfectly fine for meeting the tenets in the guidance document as it's written right now. So I don't want to leave a lot of dead air, and I do appreciate everyone's time. If you have other questions, you can certainly submit those by email, and we'll get back to you as soon as we can. But again, thank you for your time during your day, and we'll get these slides out to you as soon as we can. I'm going to hand it back over to Michael.
Thank you, Brian. Links to regulatory documents, other webinars, and more are available on our website. If you would like to attend future webinars, including updates from other regulatory agencies, you may submit a webinar topic request form, the link to which is on screen now and will be included in the slides. If you have any further questions regarding Brian's presentation or any of Sekisui XenoTech's consulting and contract research services or products, please feel free to ask them in the post-webinar survey or contact us through our website. So this wraps up today's session. Thanks to all for taking time out of your day to join us, and thank you to Dr. Ogilvie for his wonderful presentation. Have a great rest of your day.